Welcome to the Blue Marble, the Science Communication and Education Network. We lovingly call it the Scene, and this is a forum session, which is an opportunity to engage in conversations about the broad spectrum of science, application, and beyond. Today, we're going to be hearing from Rohan Shiradhonkar, PhD candidate at Northern Illinois University, and he's joined live by his students. And Rohan will be speaking on the Adventure Scientist program. So I'll be here. I'll be helping to moderate questions from the Zoom chat at the end of his slides. And just once again, please know that the meeting is being recorded. So with that, I'll have my screen off and I will let Rohan have the show.
Hello, everybody. Welcome to this forum. I am really excited to share about my citizen science initiative with all of you. Really glad to be doing this here with my students in attendance. There are about 20 of the students that I'm currently teaching over summer, and I've been doing some of the citizen science stuff with these students as well. So very excited to be telling them more about this. And since the forum was open to all of the public, I am going to welcome all of you. Those of you that are here to listen about citizen science, those of you that are here to listen about cool scientific stuff, new science updates, or those of you that are looking for opportunities to contribute to science, you are all at the right place. This forum is meant to bring us all together, irrespective of whether you have a scientific background or if you are an established scientist, or even if you have no science background but are still interested in science and are enthusiastic and interested in scientific activities. So as we begin, I'm going to quickly give you a little bit of context on this citizen science initiative by sharing with you my research story. And hopefully that gives you some context on how the citizen science initiative came to be and how this is supporting a real scientific research that is currently ongoing. So my name is Rohan. I am a PhD candidate in Northern Illinois University. And my research story begins way back when the Earth was just a ball of molten lava. Cut to this day, you can see that the Earth is visibly full of life. There are indicators that tell us that there is life existing on this planet. All of this life on our planet can be divided into two camps. One is the autotrophs. These are the organisms that can generate energy without depending on other organisms. And then we have heterotrophs. These are the organisms that feed on other organisms to get energy. And so if you think about it, when life forms on a planet, or when life formed on our planet, the autotrophs had a critical role to play. On our planet, the autotrophs generated energy either by using sunlight, we call it photosynthesis, or they generated energy by using chemical ingredients that were available on the planet. We call that chemosynthesis. And when these autotrophs formed on our planet, organisms like cyanobacteria changed the entire story. These tiny microscopic cells had the audacity to change the face of the entire planet. These cyanobacteria produced so much oxygen that it filled our atmosphere. And that led to the creation of ozone layer. Carbon dioxide levels went down, and all of this paved the way for complex life to evolve on our planet and survive over all these years. So that being said, all the lush greenery that we enjoy and the oxygen that we rely on is the courtesy of these cyanobacteria. the cells that invented photosynthesis on our planet and produce most of the oxygen. All of this greenery and the oxygen that we breathe is the courtesy of, again, one particular pigment known as chlorophyll A. This pigment is the green colored pigment in plants, trees, and cyanobacteria that plays the main role in producing oxygen by absorbing sunlight. And chlorophyll A is known to absorb a certain range of light to produce oxygen. And that range is called the visible range of light. Using this visible range, chlorophyll A can produce oxygen. And on Earth, it makes sense because Earth gets a lot of visible light from our sun. So on a planet that is full of visible light, chlorophyll A gets the luxury of dominating the biosphere in terms of oxygen production. However, when you start comparing our sun to the other stars in our galaxy, we notice that our sun that belongs to the yellow dwarf category and is a good source of visible light is actually not commonly found. The most common type of star, the majority of stars in our galaxy, belong to the red dwarf category. And the problem with these is that they mostly emit light in the infrared region and the radiations beyond. They produce a little bit of visible light, but that visible light is not abundant. So scientists ask the question, if visible light is barely available out there, can photosynthesis still work without the visible light? And that question was answered to a certain extent in the islands of Palau. Several decades ago, a scientist walking through the coastline of these islands, exploring new things on the island, came across certain sea squirts, and he started studying them. And what he found was an interesting discovery. What he didn't know at that moment is that that discovery is going to change the course of science for years to come. The scientist had discovered a new type of pigment in these microscopic organisms that he found. He discovered that the cyanobacteria called Acaryochloris marina contains pigment chlorophyll D, and chlorophyll D has the ability to absorb light beyond visible range. So chlorophyll D made it possible to absorb light in the far red region and near infrared region, and using that light, it produced oxygen in these cells. And that changed the story because now you have a life form on our planet that can perhaps utilize the light from M-dwarf stars and produce oxygen from it. So way down the road, all of that led to my research. In my research, in my PhD thesis, I'm looking at how chlorophyll D is made in these cells and how can we use this information to assess the relevance of chlorophyll D in looking for life on other planets. As part of this research, I get to study these cyanobacteria under different conditions. And by looking at their growth, by looking at their characteristics in these conditions, I try to understand the process of making chlorophyll D in these cells. I also look at the oxygen that is produced by these cyanobacteria under different conditions and try to analyze if that could be useful for us in looking for life on other planets. But all of this research, including mine, was possible because of this moment right here. Somebody decided to go to these places that were unknown to humans, and somebody decided to explore the unknown in these regions. And for that same reason, because of these explorations, a long line of research was started. A long line of research could be created which answered many questions in the long run. And my research is just one example. This is just one story. I do have one more story for you just to drive the point that I'm trying to make. Right here in Yellowstone National Park, in the 1960s, scientists scooped out a bacteria from a pool of boiling water. That bacteria was called Thermus aquaticus. What we found in this bacteria, again, changed the course of science for years to come. The bacteria produced an enzyme called Taq polymerase, which led to the technology called polymerase chain reaction. And if you're wondering what this technology is about, the polymerase chain reaction technology makes it possible for us to diagnose different diseases. It made possible for us to have COVID-19 testing swabs. It made it possible for us to do genetic testing and also to catch criminals using DNA forensics. All of that came from a pool of boiling water in a national park. And all of that was made possible because somebody went there and tried to uncover the unknown. That being said, this is the power of environmental microbiology. This is the power of looking at places where others have overlooked. And for that reason, I started to think, what if we all could do this? Not as scientists, but as hikers, as campers, as cavers, scuba divers, as climbers, as people who love the outdoors, as people who go to the outdoors, enjoy their time, and have fun. What if we could do this while having fun on our trips? And for that reason, I started the citizen science initiative called the Adventure Scientist Program. The Adventure Scientist Program came into being for empowering outdoor explorers to collect samples from remote environments. And these samples could power research in the fields of astrobiology, space biology, microbiology, environmental science, and many other fields of research depending on environment. So why does all of this matter? Why is it important to study these outdoor areas, these outdoor locations? A lot of these places can tell us about what life can and cannot do on our planet. Many researchers like to study cave environments to understand what type of microorganisms grow in those caves and what type of life adaptations go on in such environments. Many scientists study soil from desert areas, volcanic sites, etc., and study what kind of microorganisms grow in them. Many of them can be used for producing antibiotics and understanding the resistance to antibiotics. Many scientists also study coral reefs. These are the aquatic or marine environments that help us understand about climate change, climate resilience, and marine life. And many unique microbes that are found in such places can be used for producing bioplastics, can be used for biofuels, and similar research. So all of this can help us understand about evolution of life and perhaps also help us to look for life on other planets. When I say that, I want to bring your attention to many locations that exist on our planet that we call planetary analogs. These are the sites on our planet that resemble, if not entirely, then up to a certain extent, they resemble places on other planets. For example, the Lassen Volcanic National Park in California is an analog site for studying early Mars. The Craters of the Moon National Monument in Idaho is again an analog site for studying volcanic areas on Mars and Moon. Similarly, the Alaskan permafrost, we have the Yellowstone National Park, the Death Valley in California, and deep sea hydrothermal vents. These places is what are driving the current research in the search for life on other planets. Places like these help us understand the extremes of life on our planet, and by studying these extreme life forms on our planet, we can try to look for life on other planets. However, it is not always easy for scientists to access these locations. Many times, scientists are limited by the cost of fieldwork. Fieldwork can be extremely expensive. Fieldwork can be time-consuming and logistically difficult to carry out. Many times, scientists do not have prior outdoor training or experience of surviving in the wild. But then this is where the outdoor adventurers can fill the gap and bridge these limitations. Outdoor adventurers are known to reach the most extreme places on our planet. They are known to have extensive outdoor training, outdoor experience, surviving in the wild, and accessing these treacherous locations. And as you all might imagine, these outdoor enthusiasts are located and have ventured out to all corners of this planet. All the way from treacherous caves to underwater locations, to mountaintops, to glacial and Arctic areas. So how does this program work? How do we tap into the potential of outdoor adventurers and use it to connect the world of scientific research with them? The program starts by introducing these outdoor adventurers to environmental studies, the type of research that goes on in environmental studies, and then the equipment that is used for sampling at these locations. We provide them a small training on using these sampling kits. The training can be either in person or conducted virtually, so the scientist does not need to appear in front of the adventurer to perform this training. We make certain training videos that help these adventurers understand how sample collection works. And in order to make these sample collections scientifically reliable, we work with these outdoor adventurers to collect sampling permits from different national parks, different cave conservations, etc. We provide some training on how to keep your records when collecting these samples. The outdoor adventurers are provided with standardized sheets for record keeping and provided information on how to save the sample, how to store it, and then finally how to ship the sample back to the researcher. And once an outdoor adventurer has conducted all of that, they receive the recognition of being an adventure scientist for contributing to a real-world research. On the top here is a video of me demonstrating the sampling process to one of the adventurers. I cannot play the video here, but these are some of the videos that we use to train other outdoor adventurers when they go out and collect these samples. Here's another snapshot of a video that we use for demonstration purposes. The video goes over what things to do and things not to do during the sampling process. And then the adventurers are provided with these standardized sheets for collecting data, for note keeping, and these sheets help them to record all the environmental conditions, their observations, and other stuff that would be important for the person analyzing these samples. So when these samples reach the scientist can assess the sample quality. The scientists can look at other environmental conditions and then make a call on how good the sample is for analysis. And this is something that we have started recently. We are asking the outdoor adventurers now to use a head-mounted smartphone or have their GoPros or other cameras on their head while they are collecting these samples. This step can allow the scientist to have a better understanding of how these samples were collected in the wild. It can help the scientist to assess the quality and integrity of these samples. And by looking at the entire video of the sampling process, they can assess if the sampling protocol was correctly followed by the outdoor adventurers and use that information when analyzing these samples. It can also give them the idea of what sampling equipment was used, which location the sampling was performed at, and also the scientist can look at other details at these sampling locations that may not be available or may not be described appropriately in the record-keeping sheets. So with all of that, the program ensures not just a scientific connection between a researcher and an outdoor enthusiast, it also ensures that science is being performed correctly and with the standards that are set in research. Currently, we are working with different types of outdoor adventure groups all over Illinois. We are working with certain schools in different states and trying to see if the kids would be interested in performing some of these outdoor samplings for us. We are also in talks with a school in Guam, the American territory in the Pacific Ocean, because Guam has unique location and unique characteristics. And for my research, the cyanobacteria from Guam are going to be extremely useful. So we have sampled certain caves in Indiana, certain places in Wisconsin, in Illinois, and we are talking with other island communities. And we are also looking at outside of US. One of the talks that are currently undergoing are with some schools in island of Tonga. And we are trying to gather and connect all of these outdoor adventurers, outdoor enthusiasts, all together and work together to collect samples for my research. So one adventure scientist that I want to highlight today is Chris Landis. Chris has been a caver for last 15 years, and Chris has been visiting multiple caves that are north of, I would say, more than 50 caves in a year. And using all of that experience, using all of that caving knowledge, he has been helping us to collect samples from different caves. That's a picture of him from Sullivan Cave, which was in Indiana. And once we received those samples from Chris, we tried to analyze these samples, and we found certain cyanobacterial species that were residing in these caves. Currently, the analysis is underway to understand what type of cyanobacteria these are. And if these are the ones that I'm looking for, then this is a big achievement. And finally, I wanted to highlight some of the other adventure scientists that I could get permission for their faces. Some of the others, I didn't have permission for showing their pictures, but so they're not here. But some of them, we have Chris, we have, again, Jack here, and then we have John, and then some of these are just me sampling in different locations. And finally, I wanted to highlight the young adventure scientists that I'm currently working with. These are all the kids that are sitting in this auditorium today. They have been working with me over summer to collect some of the samples, look at different cyanobacterial species that are available around us, and I hope to use these samples in my research as well. And finally, I wanted to end by saying that if you are an adventurer, if you are an outdoor enthusiast, and you're looking for scientific opportunities, please feel free to connect with me and look at the website that we have set up. If you are a scientist and you're looking to expand the impact of your science by connecting with public, you are also welcome to join. And if you are just a curious person, please feel free to spread the word, and you may help other scientists and other researchers to connect with each other. So that is all. I do have certain extra information here, which is just in case if you have questions. But that was my talk. Thank you so much for joining me today, and thank you for listening.
Thank you for the wonderful session, Rohan. So audience members, please feel free to populate questions into the chat, or you can ask directly by unmuting your audio and video to speak. And so I'll start off with a bit of a comment and a question. So thanks for sharing that information about the Taq polymerase. It was kind of cool to know about its major applications that we take for granted. But my question is, what excites you most about these opportunities for data collection? So we feel your enthusiasm, but what most excites you?
The most exciting part about these opportunities to me, that's actually a two-parter answer. One is the scientific benefit for it, meaning now I can reach to 10 different places that I would never have done it on my own. And that means now I have a scientific data set that is more robust. I have a data set that is even more stronger and represents a broader set of samples. And the other part that excites me really is that there is a sense of providing benefit to the society. I'm able to connect people who had these interests in science, but they never had the opportunity to actually contribute to science because of their profession or because of where they are or because of other limitations. But now with this citizen science initiative, they're able to not only connect with science, but also contribute to science. And I feel like that makes it more meaningful, and that really drives me for it.
Thanks for that. You have a couple questions. Shrushti, would you like to ask your question? The question in the chat from Shrushti is the program available in Europe?
All right, cool. The question is the program available in Europe? So the program is kind of where you are. If you are interested and if you're interested in connecting, feel free to do so. I would really welcome people from all over the world to connect and join this program. The only limitation will be your own availability and the availability of either a scientist or an outdoor enthusiast in your location. But if you are in Europe and if you're an outdoor adventurer, you could definitely ship those samples to a researcher like me in the US, and then we are connected through this program. So there are no physical boundaries to this program. There's no limitations to this program. Well, physical limitations. But then the only thing is if you connect with a researcher, no matter where they are, or if you are a scientist and looking to connect with an adventurer, no matter where they are, the only thing is you communicate with each other, you tell them what you need, and you tell them what you expect, and then make the science and adventure happen at the same time.
Okay, thanks. Do you have another question from Candace Landau? Would you like to ask it, or would you like me to read it? I'll read it for her. For the scuba divers among us, is the sampling process very different from topside collection? And do you have a list of sites you're ideally looking for samples from?
Yes, thank you so much for asking the question, Candice. I am about to pull up my presentation again, just to give you one more insight into the answer. So moving to the, yes, marine environment. So I'm guessing, since the question was oriented towards scuba divers, I pulled up this slide that I have for underwater locations that I am interested in. The cyanobacteria that I'm looking for are ideally found in marine environment, but they're kind of found everywhere now. But initially, they were mostly found in marine environment. And in the marine environment, the top layer of cyanobacteria is always, or mostly the ones that have chlorophyll A in them. And right below them, where the visible light is all used up by these layer, and then the near infrared light is what is available to the cyanobacteria below them. So these chlorophyll D containing cyanobacteria are found under, probably under a biofilm, or maybe under a microbial mat, or maybe at a certain depth in the ocean. And when these, so these are the cyanobacteria that I'm interested in that are found at a little bit depth, or maybe under a microbial film or a microbial mat. And so in order to address the question itself, the sampling process looks pretty easy. You take your sampling kit. It will consist of a collection tube. It will consist of probably something to grip the microbial mat with, and put it in the collection tube, close the lid tightly without contaminating with anything else, and then you have your sample with you. In terms of DNA extraction, it doesn't need to happen on the field. You could just send the samples back to me, and I can do the DNA extractions in the lab. So, however, if you have a lab access to you, if you can access somebody's lab, or if you are a researcher, a scientist, or if you know somebody who can do it for you, then we could definitely ship the samples to the scientist that's perhaps closer to you, and then that DNA information can be shared with me. So the DNA extraction is optional. It is not at all necessary. The only important part here is collection of these samples. And collection of these samples, and that is what we'll be mostly focused on. Let me know if that doesn't answer the question.
No, I've got my mic turned on now. That was great. Thank you so much.
Cool, thank you.
You have a question from Shay Thorne. Hi from Vancouver. And how do you keep samples stable from the field to the lab where you may do DNA extractions? Or do you extract DNA in the field?
Yes, hi Shay. Thanks for asking the question. Shay is a friend. That question is definitely not planted. Just wanted to make that clear. So keeping the sample stable is definitely a challenge. We asked the adventure scientists to carry a cooler box with some ice in it while they're on their trip. And once these samples are collected, we ask these samples to be put into the cooler box so that they are kept in a cool condition. And as long as they are kept in a cool condition, these microbial biofilms that are collected, they are kind of stable in a cooler environment. We definitely don't want these samples out there for maybe like 10 days, 15 days, or maybe a month. So we try to get them back as soon as possible. But over the span of several days, the biofilm should be pretty much stable, and we can get all the data that we need from that.
You have another question, or let's see. Yeah, you have another question from UIUC. Do you have a list of sites from where you want to get samples? From Shriram Devata.
Oh, cool. Yeah, list of sites, not so much, but list of environments, yes, yes, and yes. One of the environments that I'm currently focused on is caves. And those of you that are familiar with caves or have been caving or have known people who cave, inside a cave, I'm interested in this region called the twilight zone. And this is the region where the chlorophyll D cyanobacteria are mostly found. So cyanobacterial samples from probably the mouth of the cave as well as from the twilight zone of a cave where the infrared light is enriched, that is the area that I'm interested in. So that means any cave location that can be accessed by outdoor adventurers is a site of interest for me. Another site of interest for me was the marine environment. So if you're ever accessing any marine location, any oceanic place, or any water body that is around you and that you think could have that enrichment of near infrared light, then these are the samples that I'm interested in. And again, so that becomes any water body, any oceanic area that is underexplored, that has limited amount of human intervention. Those are the sites that I'm looking for. So if you are in a particular location, and if you want to collaborate on this, send me your location, and I can tell you what nearby sites that I'm interested in, and then hopefully that might work out for you as well. So yeah, thank you again for the question.
You have a question from Susanna Scott. Hello from Mexico. Do you have any recommendations for when you ship the samples? Any best practices?
Yes, thank you, Susanna, for asking the question. Best practices is to, as I said earlier, collect these samples, and as soon as you collect them, put them on ice and have them shipped right away if possible. If not possible, have them shipped in a day or two. And whatever shipping time it takes, we cannot control that. But what I've noticed so far is that these samples, as long as they are in their biofilm, they are pretty intact and they're kind of stable. They do get degraded over time, but even in that situation, they can still answer the questions that we are looking for, and the data from them is still valuable.
You have another question from Arunava Podar. Is this program specifically focused on addressing research objectives related to your current PhD project, or does it have a broader scope?
Yes, let me answer that over here. So most of my collections so far have been focused on my own research, and that is because I do have to satisfy my PhD committee in terms of the data that I'm generating from this. So that is why my current focus is on my own research, but that does not limit the scope of the project, the program itself. The Adventure Scientist program is much bigger than my own research. It can contribute to your research if you are studying anything from the environment. It can contribute to anybody's research out in the entire world who is studying something from the environment. For just a random example, think about a scientist sitting in Japan who is interested in studying the waters in USA. All he has to do is look for adventurers in that region, collaborate with them. The adventurers can collect the sample, send it to Japan, and the researcher in Japan can now study waters that are in US. And that means the scope of the program isn't limited to any one particular area of interest. As I told, it depends on what the scientist is looking for. If the scientist is looking for soils from a desert region, all you have to do is find an adventurer that is closer to a desert location, or an adventurer who is willing to travel to a desert location, and then get to collaborate with them and get the samples shipped to his lab. So the scope, as far as the scope goes, it is only limited by the willingness of people and the scientific and outdoor interests of people that are involved. But for now, since this is the pilot phase of this program, we're still trying to iron out many of the problems, many of the issues, the sampling permits, things like that. I have kept the program very much focused on my own research, but it doesn't have to be. All it needs is another researcher or another scientist interested in this program, and then he can tap into the same network of adventurers that we have currently, and then utilize that network for other kind of sampling.
Luca Lowy, and I'm sorry if I mispronounced your name or any other names that I've said. Luca Lowy says, hi from UIC. In addition to caves, could abandoned mines and quarries be possible sample locations? One of the cave conservancies in the state he grew up in, Pennsylvania, has several mines under their care.
Can you read the first part of the question again? Sorry.
Sure. In addition to caves, could abandoned mines and quarries be possible sample locations?
Yes, that is an interesting premise to me. Definitely abandoned mines have a caving ring to it. And when I say that, I'm again thinking about the far red light enrichment that can happen inside a mine. I'm trying to go forward to the slide where I had the cave example here. So yes, both of those places I find them interesting to sample. And yes, this is the slide. So yeah, I'm assuming that even in those abandoned mines, we would have these twilight zones where there is not enough visible light, but there is enough near infrared light that is enriched in that location. And so having samples from those locations would be extremely exciting. And especially since those abandoned mines would have less human impact, less human interference, that gives us even a better sampling opportunity, meaning those samples would be even more meaningful in terms of the microbial population that grows in there. So yeah, thank you for the question.
Great. Aini Danish asks, she's really curious to how do you make sure the collected samples are not being cross-contaminated by the other possible microbial life or microbes around the sample site?
Yes, excellent question. And I believe that the question comes from certain experience in field sampling, because it is a sad truth of field sampling that there is hardly things that you can control when you're out in the wild. The example that I was trying to think of is right here when I was trying to sample the cyanobacteria between these rocks. The water was so violent that it would crash against the rock, and all of that water would, the droplets of water would get into my tube unexpectedly. And there was nothing that I could do to prevent that. And the harsh reality of field sampling is that there are certain things that you can definitely control and minimize, but then there are certain things that are outside your control, and you just have to account for them when you analyze the samples. So does that mean that the sample itself is unusable Not at all. Does that mean that we have to account for impurities in the sample? Totally yes. And so that is why having measures like these, having a recording of the entire sampling process can let the scientists know that there has been a contamination in your sample. So you won't be an oblivious scientist anymore, thinking that your sample is pure, because now you have access to the entire process of sampling, and you can see if something else went into the collection tube, or if the person sampling had committed some kind of error while he was collecting these samples. So having these kind of measures minimizes those errors, but then it is really hard to avoid them completely and have a zero error rate on that. So thanks for asking the question.
Thanks. And I think, to piggyback onto that, I think the metadata that you collect for the details for sampling reliability, it's so essential. And on that note, do you get to hear back from scientists for any of the assays that were used for analyses or any of the sequencing, or do you expect any reuse that can be enabled depending on the samples?
Could you read the question again? Sorry, there's some interference.
Oh, no worries. So do you get to hear back from the scientists to process assays for you?
Yes. So currently we haven't reached out to many other scientists that will be processing these samples. So most of the sample processing happens in our lab. And we just share the data with other adventurers on what have we found in these samples. But as the program grows and as other scientists join in this program, I would assume that there has to be this crosstalk between what type of sampling methods they want to utilize, what type of sampling assays they want for their samples, and that will be the call the call of those scientists. So the scientist who is going to finally process the sample gets to decide what type of sampling assays should be used, what type of sampling equipment should be used, what standards of sampling would he want from these samples, and so on. So to answer the question, currently, no. Everything is being done in my own lab, but I assume that as the program becomes bigger and as more scientists join in, we would have that type of decision-making during those steps.
Great. Well, I hope to hear progress after your PhD of this program growing. That's it for the questions that are in the chat. By any chance, do any of your students have questions they'd like to ask live?
Yeah, that's interesting. Some of the students are nodding their heads, saying no.
If you'd like to ask a question, you can hand them. Well, they can ask a question, and then you can repeat it out loud.
Yeah. If you guys have any questions, feel free to ask. But I also want to state that I have been going over most of these things with them over the last couple of weeks. The sampling images that I showed you was after a really long lecture about environmental science and some training, some demonstrations on sampling. So all the questions that they would have had, I think, have been asked already. But we do have one volunteer asking the question. I'm going to take a mic to you just to put you on the spot. Say your name and then ask the question.
My name is Xavier. Could you go back to the slide where you show the suns? The suns?
The suns?
Yeah.
Yes.
Stop. Stop.
Yes.
I was wondering if different stars produce different infrared light, or like different light, depending on their color or size.
Yes. So if I understand the question correctly, you're asking, depending on the color of the star, do they emit different type of light?
Yeah.
Okay. So the answer is yes. And to just give you one quick overview of the difference between our sun, and a red dwarf, I do have whoops, screen sharing stopped. Hold on. Screen share again. Okay, so I guess the screen share is up again. And this is the slide that I was talking about here. You are seeing two different radiation graphs. The graph on the left is from our sun. So this is the light that is emitted by our sun, a yellow dwarf. And this is the light emitted by a red dwarf. And what you're seeing here is that the range from roughly 400 to 700, that's the visible range. And you can see that there's so much light being emitted in this visible range. When you go to a red dwarf, the visible light emitted by these is there, but it's still much lesser than our sun. And then as you go farther along the radiation wavelengths, you see more and more light being emitted here. So depending on the color of the star, they emit different radiations, different amounts of light, and yes, that makes the difference between all of these stars. Thank you for asking the question. We have one more question. Pass on the mic. Is it turned on? Yes. So say your name, what grade you're in, and then ask the question.
My name Mateo, and I have a question about these stars. Why is the brown planet? Wait, can you go back to the stars, please?
Sure.
I don't know what it's called.
Guys. Yep.
So why is it called the brown dwarf star even though it's purple? And what's the light? What's the light for the blue giant stars and the white dwarf stars?
That one is kind of hard to answer for me. Maybe if there is an astronomer in the audience online, feel free to answer that question. For me, I have very limited information on the other type of stars that are in our galaxy. The question that why is it called a brown star even though it looks purple is actually beyond me. So the way these people name them is for ease of understanding and to indicate something about these stars, but it doesn't really reflect on the light that is coming from these stars. So yeah, I wish I had a better answer for you, but I do not. We have one more question.
Since we only focused on chlorophyll A and chlorophyll D, isn't there like other chlorophyll? Since we only focus on those two.
I love that question because when I do these presentations in my department, I always have to answer that. And that would tell you that I would be better prepared to answer this question, which I am not. But yes, there are other types of chlorophyll that exist in the nature. The reason why I am focusing on chlorophyll A and D is because these play the main role in producing oxygen, which helps the other life on our planet. But then there are also chlorophyll F that is important in such situations. Chlorophyll F can help chlorophyll A produce the oxygen. So chlorophyll can absorb some of the infrared light, the near-infrared light, and provide that energy to chlorophyll A. But chlorophyll A is, again, the main pigment that produces the oxygen. And then there's chlorophyll B, there's chlorophyll C, there's chlorophyll E, F. One of them, I think, hasn't been discovered yet. It has been made in a lab, but not discovered in the environment. But yeah, there are other types of chlorophylls, and each type of chlorophyll absorbs light differently. So depending on their absorption spectrum, these were named, and these were named in the sequence that they were discovered. So chlorophyll A was the first chlorophyll to be discovered, so it got the name A. And then the next one to be discovered was chlorophyll B. The next one, I think, was synthesized in the lab. And so as they started discovering these, they started naming these A, B, C, D, E, and so on. So, yes. Thank you for the question.
You have a final question in the chat, and we'll close out with this one from
Okay.
Theophilus Eshun. Some microbes do not need light for photosynthesis. What about those ones?
Yes, interesting question. Thank you for pointing that out. Many microbes do not depend on visible light. Well, even if they depend on visible light, they do not produce oxygen in that type of photosynthesis. So my research focuses on very niche area of photosynthesis that produces oxygen. And the technical term for this is oxygenic photosynthesis. So as far as oxygenic photosynthesis is concerned, that these pigments are extremely important. But if you remove the oxygen from the equation, yes, there are other photosynthetic organisms that can absorb light and generate energy from it. The only difference is they do not produce oxygen, and hence my research is not focused on them. But there are many researchers out there that are discovering the awesome and amazing characteristics of those photosynthetic organisms. And yes, that is another area of research that could benefit from the Adventure Scientist program.
Great. Thank you again for your time, Rohan. Thank you, students, for accompanying us, and thank you to our live audience who participated. So we appreciate all of your time and contributions, and we look forward to a future forum session. So thank you again. Please join me in a round of thanking Rohan.
Thank you so much.